So this lecture will be on protein degradation. This is the usual disclaimer that it is meant for you people only during this COVID-19 forced lockdown, and it should not be used for any other purpose, copied or posted on any site without the permission of the author. And some of the pictures in the presentation have been taken from public domain websites or popular books, or for that matter, some journals. So so that the topic can be easily uh, the topic becomes easy to understand. The credit for such pictures remains with the original publisher, right? Okay, so contrary to our belief, this protein degradation is also a highly organized activity, something similar to what we have seen in case of protein synthesis. You have seen in details how proteins are synthesized in a very organized, in a very structured manner. Similar to that, protein synthesis is also an organized activity, and it is carried out by sophisticated cellular and molecular machinery which is present in the cell. And we started learning about this machinery, which degrades, which is responsible for degradation of the protein uh, slowly. Now the next question that comes, what is the need? What is the rationale behind this protein degradation? So please understand that protein profile of a cell changes with the changing in the surrounding condition. So when the condition surrounding a given cell or an organism changes, the protein profile changes. Some of the proteins are required more than the other proteins under a given set of conditions. So proteins which are required more, their synthesis is upregulated, and those which are not required at that point of time, their synthesis is downregulated, and the existing protein, they are diverted toward degradation. So this protein degradation therefore is necessary for adaptation to changes in surrounding conditions. So depending upon the surrounding condition, protein profile changes, and accordingly it helps in adapting the cell or the organism to the surrounding condition. Similarly, this degradation helps eliminate abnormal proteins, mutant proteins, or proteins which are not properly folded, misfolded proteins, because these proteins won't have the desired biological activity, and therefore it is better to get rid of them, okay, and synthesize a fresh copy. So protein degradation helps eliminate those abnormal, mutant, or misfolded proteins. Similarly, proteins undergo oxidative damage over a period of time because of their continuous usage. They may undergo some damage, and therefore they may need to be replaced. So for this purpose also, protein degradation is necessary. Okay, so proteins which are meant for degradation in the cell, they are labeled or tagged with ubiquitin. So ubiquitin is a small molecular weight protein. Ubiquitin is a small molecular weight protein. Molecular weight is only 76 kilodalton. It is made up of only 76 amino acids. Okay, and it is highly conserved. A very characteristic feature of this protein is that it is highly conserved. Okay, what does it mean? It means that over a period of time, it has not undergone many changes in its amino acid sequence. Similarly, the ubiquitin which is present in yeast and ubiquitin which is present in humans, they differ in amino acid sequence with respect to amino acid sequence only in 3 amino acids. 3 amino acids are different in yeast ubiquitin as compared to the amino acid sequence of the ubiquitin which is present in humans. Now this ubiquitin, it is added to the target protein by a series of enzyme catalyzed reactions. So as I said in the beginning itself, that it is a highly structured activity. So this ubiquitin is added to the target protein, the one which needs to be degraded, target protein, by a series of enzyme catalyzed reactions. And it binds to the target protein by an isopeptide bond. Okay, please remember, by an isopeptide bond. And this isopeptide bond is formed between amino acid glycine of ubiquitin, which is present at the C terminus, and amino acid lysine of the target protein. Okay, a specific lysine of the target protein and C-terminus glycine of the ubiquitin. These are involved in the formation of isopeptide bond. So using this isopeptide bond, this ubiquitin is added, is tagged, is labeled to the target protein. Now let us understand what exactly is meant by isopeptide bond. So a peptide bond is formed between the carboxyl group and amino group, you know. But what is important is that this carboxyl group and amino group, both these groups should be attached to alpha carbon atom of the respective amino acids. Then such a bond is called as peptide bond. For example, this is amino acid number 1, R1. Okay. And this is amino acid number 2, R2. And this is alpha carbon. And this is also alpha carbon. Okay. So carboxyl group attached to alpha carbon and amino group attached to alpha carbon. You can also see amino group attached to alpha carbon, this, and carboxyl group attached to alpha carbon. So peptide bond will be formed between these two. Okay, this is the peptide bond. This is called as peptide bond. And what is an isopeptide bond? It is also formed between carboxyl group and amino group. But the difference is that carboxyl group is contributed by alpha carbon. This is carboxyl group contributed by alpha carbon. And the amino group is contributed by an epsilon carbon. This is alpha carbon. This is beta. This is gamma. This is delta. And this is epsilon. So this amino group is contributed by epsilon carbon, and a peptide bond is formed between these two. The bond remains the same, but the groups are contributed, the functional groups are contributed by different carbons. Carboxyl group is contributed by alpha carbon, and amino group is contributed by epsilon carbon. This is amino acid lysine. So amino acid lysine has a epsilon amino group. Okay, so it is involved in the formation of peptide bond. Bond remains the same. This is the bond. But the thing is, the groups are contributed by alpha carbon and epsilon carbon respectively, and therefore this is called as an isopeptide bond. Okay, so please remember the difference between a peptide bond and isopeptide bond. Whereas in case of peptide bond, both the functional groups, carboxyl as well as amino, they are contributed by alpha carbon. In case of isopeptide bond, carboxyl group is contributed by alpha carbon and amino group is contributed by epsilon carbon. Therefore, it is called as an isopeptide bond. Okay, so how this ubiquitin is added to the target protein? So as I said, it is added to the target protein by a series of enzyme catalyzed reaction. So there are 3 enzymes. You know, in some books they are mentioned as E1, E2, and E3. In some other books, their names are also mentioned. E1 is called as ubiquitin activating enzyme. E2 is called as ubiquitin conjugating enzyme. And E3 is called as ubiquitin protein ligases. Okay. So what is the reaction? In reaction 1, ubiquitin is transferred to a specific cysteine residue of enzyme E1. Okay, I'm repeating it. Ubiquitin is transferred to specific cysteine, CYS, residue of enzyme E1. E1 is ubiquitin activating enzyme. And they are bonded by an thioester bond. And this is an ATP driven reaction. ATP is involved over here. ATP hydrolysis takes place and 2 high energy bonds are utilized in this particular reaction. In reaction 2, ubiquitin from enzyme E1 is transferred to another specific cysteine amino acid of enzyme E2. Again, the bond is a thioester bond. This is what is called as a thioester bond. Okay, so ubiquitin from enzyme E1, from cysteine amino acid cysteine of enzyme E1 is transferred to a specific cysteine of enzyme E2. The bond is a thioester bond and the enzyme is ubiquitin conjugating enzyme. And in reaction 3, which is catalyzed by ubiquitin protein ligase E3, this is reaction 3. ubiquitin from enzyme E2 is transferred to specific lysine of target protein. This is the target protein. A specific lysine will be contributing a epsilon amino group, and ubiquitin with its C-terminus carboxyl group, it will form an isopeptide bond. This is an isopeptide bond. So it will be transferred to target protein, and the bond involved will be an isopeptide bond. The reaction is catalyzed by enzyme E3, which is ubiquitin protein ligase. Okay. So whereas we have only one enzyme E1 and enzyme E2, we have multiple enzyme E3. We have good number of E3 enzymes. In fact, there is a whole family of E3 enzymes present in the cell. They are ligases, and they provide the much needed substrate specificity. What does it mean? It means that there is a specific ligase either for one protein or a group of similar proteins. That particular ligase will be transferring ubiquitin to the target protein. Okay, so you have a specific E3 enzyme either for an individual protein or a group of similar proteins, and this enzyme will be responsible for labeling these proteins, with the tag that is ubiquitin. Okay, so therefore you have a whole family of ubiquitin protein ligases. Okay, so I'm repeating it once again. So far as we know, only E1 and E2, they may be just one type of E1 and E2, but we have multiple types of E3. Okay, and these multiple types of E3s, they provide the much needed substrate specificity, because you have a diverse array of proteins which need to be tagged. Okay, and each protein or a group of similar proteins will be tagged by one particular E3 enzyme, ubiquitin protein ligase. It will be specific for them only. Okay, so proteins have been tagged. What next? The question remains, who will carry out the degradation? Okay, so who will execute the degradation? So there is what you can say, there is a proteasome complex. This is called as the executioner. So tagged ubiquitinated protein, ubiquitin tagged, they are also called as ubiquitinated proteins, are handed over to proteasome complex for degradation. Proteasome complex will execute the degradation. It requires ATP, this degradation process. It also requires ATP the way protein synthesis require ATP. Similarly, another characteristic is this proteasome complex, it does not digest ubiquitin. Ubiquitin is not digested here. Okay, ubiquitin is a tag, so it is not digested. This proteasome complex is a macromolecular assembly. It has a molecular mass of close to 2000 Dalton. So it is almost as big as bacterial ribosome. So bacterial ribosome is 2700 kilodalton. This proteasome, which is present in eukaryotic cell, it has a molecular mass of close to 2000 kilodalton. And structurally, it is made up of two components. One is what is called as a catalytic component, which is 20S, and the other one is called as a regulatory component, which is 19S. Okay, and each one of them in turn, for example, catalytic component in turn is made up of 14 alpha subunits, 14 alpha subunits and 14 beta subunits. So proteasome is made up of catalytic component and regulatory component, and catalytic component in turn is made up of 14 alpha subunits and 14 beta subunits. Similarly, regulatory component, it is made up of 20 subunits. So therefore, you can imagine such a high molecular mass, right? This regulatory component, 19S, it has 6 distinct ATPases of AAA class. 6 distinct ATPases, ATP hydrolyzing enzymes, ATP hydrolyzing activities of AAA, AAA class, ATPase associated with various cellular activities. A of ATPase, 1A, associated second A, and activities third A. Therefore, they are called as AAA class. ATPases associated with various cellular activities. So it has got 6 distinct ATPases activity sites. Similarly, This 19S regulatory component also has an isopeptidase activity, or it has an isopeptidase enzyme also, which is responsible for cleaving the isopeptide bond by which ubiquitin is tagged to the target protein. So isopeptide bond will be cleaved and ubiquitin will be recycled back. It is not digested. Please remember, proteasome is not capable of digesting. It does not digest ubiquitin. That isopeptide bond will be cleaved, will be hydrolyzed, will be broken by a specific isopeptidase, which is a part of 19S regulatory component, and ubiquitin, the resulting ubiquitin, will be recycled back. Let us see the structure of these in little more details. So the subunits of the catalytic component, you remember now there are 14 alpha and there are 14 beta subunits. So the subunits of catalytic components are arranged in 4 rings of 7 subunits each, one above the other in the form of a barrel. So these are the 4 rings. This is one ring to second ring, third ring, and 4 rings. And they are placed one above the other. Each ring is made up of 7 subunits, okay? And they are arranged in such a way that they form a barrel-shaped structure. The upper and lower rings are of alpha subunits. This ring is of alpha subunit, 7 of them, and this is also made up of alpha subunit, 7 of them. While the 2 inner rings, these 2, they are made up of beta subunits. There will be 7 here and there will be 7 here, right? Similarly, the active sites of proteases are believed to be located on the N termini of certain beta subunits. It is believed that there may be 6 different types of proteolytic activity, and those active sites of proteases are located on the N termini of certain beta subunits, inner rings. Some of the beta subunits will have active sites of those proteases. The barrel, this barrel-shaped structure is sealed on either side by 10 subunits each of 19S regulatory component on both ends. So this is 10 subunits of the regulatory component, and this is another 10 subunit of the regulatory component. So this barrel will be sealed from both the sides by 10 subunits of regulatory component and 10 subunits each of regulatory component, right? Now polyubiquitinated proteins, okay? Just one ubiquitin, serves as a very, what you can say, a mild signal for degradation. So there has to be minimum 4 ubiquitins, then it becomes a strong signal. So therefore we call it as a polyubiquitinated protein. So polyubiquitinated protein with minimum 4 or more than 4. So this becomes a strong signal for degradation. Addition of one ubiquitin will be just a mild signal. Okay. So minimum 4 ubiquitins are tagged or more than 4 ubiquitins are tagged. So this polyubiquitinated protein, they are sequestered. Their entry into this barrel is regulated. Regulated by whom? By this regulatory subunit. They are sequestered through the core of the barrel-shaped proteasome. Okay, and their entry is regulated by regulatory component. Polyubiquitinated proteins with minimum 4 ubiquitins tagged to them, they will be allowed to pass through this barrel, through the core of this barrel, and their entry will be regulated by the regulatory component. Okay, and now you know that this regulatory component has a isopeptidase also, so that isopeptidase will cleave that ubiquitin, and those ubiquitins, those liberated ubiquitins, they will be recycled back. Proteins in the barrel, in the core of the barrel, they will be digested processively. Okay, because this beta subunit, it has got, as I said, it is believed to have as many as 6 different types of proteolytic activities, and these proteolytic activities are located at the N termini of certain B subunits. There are 14 of them. So some of the beta subunits at the N terminus, they have proteolytic active sites. So those proteolytic active sites, they will be processively digesting the protein which is passing through it until what time? Until they are reduced to peptides ranging in length between 7 to 9 residues. So they will be digested processively until what time? Until they are reduced to peptides ranging in length between 7 to 9 residues. So those peptides will come out from this end, right? So ubiquitinated protein will enter from this end, and after digestion, after they have been reduced to peptides ranging in length between 7 to 9 residues, they will come out from this end. So for example, so this is 20S proteasome. As you can see, a beautiful picture. So there are two outer rings which are made up of alpha subunits. And the two middle rings, the one in yellow, they are catalytically active beta sites, beta subunits. Okay. And some of these beta subunits, they have the proteolytic activity, the active centers of the proteases, right? And each ring is made up of 7 subunits. You can count 1, 2, 3, 4, 5, 6, 7. Each ring is made up of 7 subunits, and they are placed one above the other. And then this barrel-shaped structure, which is made up of catalytic component, is sealed on both hands by this regulatory component. There are 10 subunits on this hand. This is the regulatory component, the one in red, and this is the regulatory component. Okay. And it has got an isopeptidase also. So what does it do? It will cleave that isopeptide bond. This is a polyubiquitinated protein. Okay, you can see there are more than 4 ubiquitins. Okay, 4 or more than 4 ubiquitins serves as a strong signal. So this polyubiquitinated protein will make its entry into this barrel-shaped proteasome. Their entry is regulated by this 19S regulatory subunit. Okay, and it has got a isopeptidase which will cleave the ubiquitin, which will cleave that isopeptide bond, and this ubiquitin will be recycled back. And the rest of the protein will pass through the core of this barrel-shaped structure. Please remember, this beta subunit has got proteases active sites. So those proteases will act on that protein till the time they are reduced to peptides ranging in length between 7 to 9 amino acids. Okay, and these peptides, these short peptides, they will be freed, they will come out of this barrel-shaped structure from the other end, right? Now, this half-life of protein, it is proteins, they come stamped with the expiry date. At the time of synthesis itself, you know, their half-life has been decided. Okay, and accordingly, proteins will perform, they will carry out their function, and accordingly, they will be degraded, they will be turned over. Okay, half-life of a protein is determined by several factors based on its structure and primarily function. What kind of function, what kind of role it is, it will play or it plays in the metabolism. Based on that, their half-life is determined. For example, you know, you can have two broad categories. You can have proteins which are long-lived and proteins which are relatively short-lived. So under long-lived proteins, as a general rule, housekeeping proteins, those which are involved in routine functions, they generally have longer half-lives. They are not turned over very frequently. Similarly, lysosomal proteins. So now you know lysosome is home to as many as 20 different types of acid hydrolases. So those proteins, they generally have longer half-lives. Enzyme proteins which are catalyzing non-regulatory steps in metabolic pathway, even they have relatively longer half-lives. On the other hand, regulatory proteins which are involved in catalyzing regulatory steps in metabolic pathway, they are generally short-lived. Transcription factors, in eukaryotes, you have large number of transcription factors, you know, which are involved during the process of transcription. So those transcription factors, they are generally short-lived. Proteins involved in cell division and growth, they are short-lived. And proteins with PEST sequence, PEST means having proline, glutamic acid, serine, threonine. This is one letter, what you can say, designation for amino acids. So proteins having this sequence, PEST sequence, they are also generally short-lived. Half-life of protein, it is also determined by the N-terminal amino acid. A very simple signal determining the half-life of proteins is its N-terminal amino acid. What is there at the N-terminus? Even that gives you some idea as to what is going to be the half-life of this particular protein. And this is referred as N-terminal rule. And according to this N-terminal rule, there are three major categories of proteins. And what are those three categories? Highly stabilizing means proteins where the half-life is more than 20 hours. More than 20 hours. Okay. That is one category. We are calling it as a highly stabilizing. And such proteins at their N-terminal will have these amino acids: alanine, cysteine, glycine, methionine, proline, serine, threonine, or valine. So if any one of these amino acids is present at the N-terminus, that protein is likely to have half-life more than 20 hours, right? The second category is called as intrinsically destabilizing. Okay, means these proteins will have half-life anywhere between 2 minutes to 30 minutes, right? So they are short-lived proteins. So if the N-terminus has amino acid arginine, histidine, isoleucine, leucine, lysine, phenylalanine, tryptophan, or tyrosine. So these three aromatic amino acids. Okay. So such amino acids are present. If any one of these amino acids is present at the N-terminus of a given protein, then it is a signal, it is an indication that this protein will have a half-life in the range of 2 to 30 minutes. And the third category is destabilizing after chemical modification. So intrinsically they are not destabilizing, but if chemical modification takes place, it becomes destabilizing. Such proteins will have a T-half of in the range of 3 to 30 minutes, half-life of anywhere between 3 to 30 minutes. So what are those proteins? What are those amino acids? Asparagine. Okay, if aspartic acid is there, it may not have, but if it gets chemically modified to asparagine, then it becomes destabilizing after chemical modification. Similarly, aspartic acid, if asparagine is there originally, it may not be, but if it gets chemically modified to aspartic acid, it will become destabilizing. Similarly, glutamine and glutamic acid. So if these amino acids are there, modified version of their original one, then they become destabilizing. We are calling it as destabilizing after chemical modification. So if these amino acids are there at the N-terminus and they have arrived because of a chemical modification, then such proteins will have a half-life of 3 to 30 minutes. Other signals are also there, but less understood. So other signals which might help identify proteins for destruction include cyclin destruction box. If it is present, then it is also a signal that this protein is ripe for degradation. Similarly, as I said earlier, proteins with PES sequences, proline, glutamic acid, serine, threonine, these proteins are also generally short-lived. So what is the significance of protein degradation? So it is being used for regulating a number of biological functions. So now you can understand also, you can visualize also that this controlled degradation of protein, depending upon the surrounding situation, maintaining the protein profile of the cell, it can help regulate a number of biological functions. It helps in regulation by dynamically altering the abundance and stability of key regulatory proteins. Okay, dynamically altering the abundance means copy number of those proteins and stability of key regulatory proteins. Activation of specific E3 ligases. E3 ligases means E3 enzymes has been observed in more than 90% cases of cervical carcinoma type of cancer. So what happens, which is caused by virus? This virus does what? Viral infection activates enzyme E3. You know enzyme E3, which is a protein ligase, E3 protein ligase. And what does it do? This activated enzyme in turn, tags tumor suppressing proteins in the cell, proteins which are involved in DNA damage repair. Those proteins are tagged by this activated E3 enzyme. Okay. And because these proteins have been tagged with ubiquitin, they undergo degradation. What proteins? Tumor suppressing protein. What proteins? Proteins which are involved in DNA damage repair. All those proteins are prematurely tagged because of activation, untimely activation of enzyme E3. This enzyme got activated because of a viral infection. Okay, so tumor suppressor proteins have been tagged, DNA damage repair proteins have been tagged untimely. When it was not required, that tagged with ubiquitin and they undergo degradation. So these proteins are no longer available in the cell, and as a result of that, the cell becomes prone for unregulated growth, resulting in cancer. Similarly, now it's a hot topic also. Similarly, ubiquitin proteasome pathway, that the one that we have just discussed, it is now believed to be involved in mounting an appropriate cellular response against abiotic stress in plants. Plants, they undergo, they face different types of abiotic stress. It may be temperature, it may be salt, it may be water. So ubiquitin proteasome pathway involving E3 enzyme again, it is now believed to be responsible for mounting a suitable cellular response against a specific type of abiotic stress. And let me tell you that this is a very hot topic as far as research is concerned. Some of the biological processes thought to be regulated by protein degradation include, I have given, this is not an exhaustive list, some of the biological processes which are regulated by protein degradation: gene transcription, cell cycle progression, inflammatory response, tumor suppression, cholesterol metabolism, antigen processing, circadian rhythm. The list is not exhaustive, but you can remember some of them. These are some of the biological processes which are regulated by protein degradation. So protein degradation has its own significance as far as cellular metabolism is concerned. So that's all I have in this particular lecture. Thank you very much.